From our previous lecture, we have talked about the two major categories of microbes, which are cellular microbes or the microorganisms and the acellular microbes or the infectious particles. So today, we will learn about the structure of the organisms. So because they are so small, and for us to see their structures, we have to make use of the compound light microscope. But to further view the ultrastructures of microbes or the tiniest parts of it, we have to make use of the electron microscopes. Also, in this lecture, we will be discussing about the ways in which microbes and their cells reproduce and how microorganisms are classified. We all know from biology that cell is the fundamental unit of any living organism. So a cell obtains its food or nutrients from the environment to produce energy for metabolism and other activities. Metabolism refers to all of the chemical reactions that occur within a cell, and because of its metabolism, a cell can grow and reproduce. So it can respond to stimuli in its environment such as light, heat, cold, and the presence of chemicals. It can also mutate or change its DNA makeup for it to become better suited for its environment, to adapt and to survive. Cells are classified into prokaryotes and eukaryotes. Prokaryotes are simpler cells. They do not have complex system of membranes and organelles. Under the prokaryotes are bacteria and archaea. Eukaryotes, on the other hand, are more complex cells. They contain true nucleus and many membrane-bound organelles. Under the eukaryotic cells are algae, protozoa, fungi, plants, animals, and humans. Now we'll talk about the eukaryotic cells. Eukaryote comes from words eu or true and karyo or not or nucleus, because they have a true nucleus and that their DNA is enclosed by a nuclear membrane. Most animal and plant cells are 10 to 30 micrometers in diameter, about 10 times larger than most prokaryotic cells. This is an example of an animal cell with all its structure and organelles in it. These are the structures of a eukaryotic cell. We will discuss them one by one in detail. The cell is enclosed and held intact by the cell membrane, which is also known as the plasma membrane or the cytoplasmic membrane. Structurally, it is a mosaic composed of large molecules of proteins and phospholipids. The cell membrane is like a skin around the cell, so it separates the contents of the cell from the outside world. Cell membranes have selective permeability, meaning that they allow only certain substances to pass through them. The nucleus. This is the primary difference between prokaryotic and the eukaryotic cell. Eukaryotic cells possess a true nucleus, whereas prokaryotic cells do not. The nucleus controls the function of the entire cell and can be thought of as the command center of the cell. It has three components: the nucleoplasm, the nuclear membrane, and the chromosomes. Nucleoplasm is the gelatinous matrix. The membrane that serves as the skin around the nucleus is called the nuclear membrane. It contains the holes or nuclear pores through which large molecules can enter and exit the nucleus. The chromosomes, or the eukaryotic chromosomes, are the ones that are embedded or suspended in the nucleoplasm. They consist of the linear DNA molecules and proteins. Genes are located along the DNA molecules. So each gene contains the genetic information that enables the cell to produce one or more gene products. The cytoplasm. It is a semi-fluid gelatinous nutrient matrix. So within the cytoplasms are organelles. These organelles are storage granules. It includes the endoplasmic reticulum, the ribosomes, Golgi complexes, mitochondria, centrioles, microtubules, lysosomes, and other membrane-bound vacuoles. So each of these have highly specific function, and all of the functions are interrelated to maintain the cell and allow it to properly perform its activities. The cytoplasm is where most of the cell's metabolic reactions occur. We will discuss now the contents of the cytoplasm. First is the endoplasmic reticulum. The ER is a highly convoluted system of membranes that are interconnected and arranged to form a transport network of tubules and flattened sacs within the cytoplasm. Much of the ER has a rough, granular appearance when observed by transmission electron microscopy. This rough appearance is caused by the many ribosomes attached to the outer surface of the membranes. Ribosomes that are not attached is called the smooth endoplasmic reticulum. Ribosomes. Eukaryotic ribosomes are 18 to 22 nanometer in diameter. They consist mainly of ribosomal RNA and protein, and they play an important part in the synthesis or manufacture of proteins. Within the cell, the ribosomes are the size of protein synthesis. But most of the proteins released from the ER are not mature enough, so they must undergo first further processing in the Golgi complex before they'll be able to perform their functions. Ribosomes can be freely floating or they can be attached in the rough endoplasmic reticulum. The Golgi complex. This is also known as the Golgi apparatus or Golgi body. This connects or communicates within the ER. This stack of flattened membranous sacs completes the transformation of newly synthesized proteins into mature, functional ones and packages them into small membrane-enclosed vesicles for storage within the cell or export outside of the cell. So, Golgi complexes are sometimes referred to as the packaging plants. Lysosomes and peroxisomes. Lysosomes originated from the Golgi complex. They contain the lysozymes and other digestive enzymes that breaks down the foreign materials taken into the cell by phagocytosis or the engulfing of large particles by amoebas, for example, and certain types of white blood cells called the phagocytes. These enzymes also helps in breaking down worn out parts of the cell or through a process called the autolysis. When the cell sense that it is damaged or deteriorating, it will initiate cell death. Peroxisomes naman are membrane-bound vesicles in which hydrogen peroxide is both generated and broken down. Peroxisomes are found in mammalian liver cells. The mitochondria. This is called the powerhouse of the cell. This is very important because in the mitochondria, the ATPs are formed by cellular respiration. ATPs, or the adenosine triphosphate, are very important for the cell to function. Without it, the cell will not be able to perform its function. So in this process, the energy is released from the glucose molecules and other nutrients to drive other cellular functions. Plant cells and algae contain both mitochondria and another type of energy-producing organelle, which is the plastid. Plastids are membrane-bound structures containing a lot of photosynthetic pigments. Chloroplast, one type of plastid, contains a green photosynthetic pigment called the chlorophyll. So, as we all know, photosynthesis is a process by which light energy is used to convert carbon dioxide and water into carbohydrates and oxygen. Thus, photosynthesis converts light energy into chemical energy. Throughout the cytoplasm is a system of fibers collectively known as the cytoskeleton. There are three types of cytoskeletal fibers. They are the microtubules, the microfilaments, and the intermediate filaments. All these types of cytoskeletal fibers serve to strengthen, support, and stiffen the cell and give the cell its shape. Microtubules are slender hollow tubules. Microfilaments are slender thread-like contractile structures which facilitate cell contraction. In anatomy and physiology of skeletal muscles, we have seen the importance of this structure in muscle contraction. So without the cytoskeleton, these fibers, we will not be able to produce muscle contraction. Some eukaryotic cells contain cell walls. These are the external structures that provide rigidity, shape, and protection to the cell. When it comes to structure of cell walls, eukaryotic cells have a much simpler structure than prokaryotic cell walls. They may contain cellulose, pectin, lignine, chitin, and some mineral salts. The cell walls of algae contain a polysaccharide called the cellulose. This is not found in the cell walls of any other microorganisms. Interestingly, the cell walls of fungi contain polysaccharide chitin, something again that is not found in the cell walls of other microorganisms. Chitin is similar in structure to cellulose, which is also found in the exoskeletons of beetles and crabs. To summarize, cell walls are present in plants, algae, fungi, and most bacteria, and are absent in animals and protozoa. Flagella and cilia are both organelles of locomotion. In some eukaryotic cells, like the spermatozoa and other types of protozoa and algae, has a relatively long thin structures called the flagella. So the whipping motion of the flagella enables flagellated cells to swim through liquid environments. Flagella are said to be whip-like. On the other hand, cilia are also organelles of locomotion, but they tend to be shorter or more hair-like, thinner, and more numerous than flagella. They can be found in some species of protozoa called the ciliates and other types of cells in our bodies, like the ciliated epithelial cells that line the respiratory tract. And unlike the flagella, cilia tend to beat in a coordinated rhythmic movement. Now we'll move on to the prokaryotic cells. Prokaryotic cells are about 10 times smaller than the eukaryotic cells. Structurally, prokaryotes are very simple cells compared with eukaryotes, and yet they are able to perform the necessary processes of life. Reproduction of prokaryotic cells is by binary fission. It is the simple division of one cell into two cells after DNA replication and the formation of a separating membrane and cell wall. All bacteria and archaea are prokaryotes. This is a typical prokaryotic cell. So as you can see, the structures are way simpler than the eukaryotic cell. These are the structures of a prokaryotic cell. Enclosing the cytoplasm of a prokaryotic cell is the cell membrane. This has a similar structure and function to the eukaryotic cell membrane. It also consists of proteins and phospholipids and are selectively permeable. Something notable about a prokaryotic cell membrane is the presence of mesosomes. These are the inward foldings of the cell membrane where cellular respiration takes place in bacteria. So this process is similar to that which occurs in the mitochondria of eukaryotic cells, where nutrients are broken down to produce energy in the form of ATP molecules. The prokaryotic chromosome usually consists of a single, long, supercoiled circular DNA molecule which serves as the control center of the bacterial cell. It is capable of duplicating itself, guiding self-division, and directing cellular activities. In prokaryotic cell, it contains no nucleoplasm and no nuclear membrane. The chromosome is suspended or embedded in the cytoplasm. Plasmids may or may not be present in bacterial cells. So a bacterial cell wall may not contain any plasmids, or it may contain one plasmid, or a multiple copies of the same plasmid, or more than one type of plasmid. So plasmids are small circular DNA molecules, double-stranded DNA that are not part of the chromosome. So they may contain 10 to 100 genes. The semi-liquid cytoplasm of prokaryotic cells consists of water, enzymes, dissolved oxygen, waste products, essential nutrients, proteins, carbohydrates, and lipids. This is a complex mixture of all the materials that the cell requires for its metabolic function. It doesn't have any organelles. The tiny particles in the bacterial cytoplasm are the cytoplasmic particles. Most of these are ribosomes. They are occurring in clusters called the polyribosomes or polysomes. Prokaryotic ribosomes are smaller than eukaryotic ribosomes, but their function is the same. They are the sites of protein synthesis. Bacterial cell walls are rigid exterior cell wall that defines the shape of the bacteria. They are chemically complex. They are made up of complex macromolecular polymer known as the peptidoglycan, or also known as murine. Peptidoglycan is only found in the bacteria. The thickness of the cell wall and the exact composition vary with the species of bacteria. Some certain bacteria are called gram-positive bacteria. They have a characteristically thick layer of peptidoglycan that is combined with thioic acid and lipothioic acid molecules. The cell walls, on the other hand, of gram-negative bacteria have a much thinner layer of peptidoglycan, but this layer is covered with a complex layer of lipid macromolecules. These are usually referred to as the outer membrane. For us to identify whether a bacteria is gram-negative or gram-positive, we use the gram staining. This is the most widely used procedure for staining bacteria. This is developed by Dr. Hans Christian Gram over a century ago. In this picture, you can see the difference between a gram-negative and a gram-positive cell wall. So, the relatively thin gram-negative cell wall contains a thin layer of peptidoglycan, an outer membrane, and lipopolysaccharide. On the other hand, the thicker gram-positive cell wall contains a thick layer of peptidoglycan and thioic and lipothioic acids. This is how we do the gram staining. First, we get a smear of bacteria and smear it into a slide. We air dry it, then heat fix by passing it through a flame a few times. Then we'll proceed with step 1. We'll add about 5 drops of crystal violet. Then we let it stand for 1 minute. The bacteria will stain purple. We wash it briefly with water and shake off the excess. Then we will proceed to step 2. We'll add about 5 drops of iodine solution to the culture. Then we let it stand for about 1 minute. Then we wash it briefly with water and shake off the excess. Then we'll proceed to step 3. So we tilt the slide and decolorize with solvent, an acetone alcohol solution, until purple color stops running. Then we immediately wash it off around 5 to 10 seconds. Then we shake off the excess water. Then we will proceed with the last step. We add 5 drops of safranin, let it stand for 1 minute, and wash it briefly with water and shake off the excess water. Then what we will see, if the bacterial stain is gram-positive, it will turn purple. If it's gram-negative, it will turn red. Some bacteria have a thick layer of material located outside their cell wall. These are the glycocalyx. Glycocalyx is a slimy gelatinous material produced by the cell membrane and secreted outside of the cell wall. There are 2 types of glycocalyx, the slime layer or the capsule. The slime layer is not highly organized and not firmly attached to the cell wall. So slime layers enable certain bacteria to glide or slide along solid surfaces and seem to protect bacteria from antibiotics and desiccation. One example of bacteria that has a slime layer is the Pseudomonas. We don't want Pseudomonas in the hospitals. These are very hard to treat kind of infection. The other type of glycocalyx is called the capsule. It is highly organized and firmly attached to the cell wall. Example of bacteria that are encapsulated are Haemophilus influenzae, the Klebsiella pneumoniae, the Neisseria meningitis, and Streptococcus pneumoniae. Flagella are thread-like protein appendages that enable bacteria to move. Flagellated bacteria are said to be motile, whereas non-flagellated bacteria are usually non-motile. Take note that bacteria never possess cilia, just flagella. There are four types of flagella arrangement in bacteria. First is monotrichous, or one flagellum. Second is lophotrichous, or a top of flagella at one end. Another is amphitrichous, or one or more flagella at each end. And peritrichous, or flagella all over the surface. This picture shows some examples of flagella. A lophotrichous flagella from the Pseudomonas aeruginosa and amphitrichous flagella on the Spirillum species. Pili and fimbriae are organelles of attachment. They enable bacteria to adhere to surfaces. They are hair-like structures more often observed in gram-negative bacteria. There are two types of pili. So the pili that enable transfer of genetic material such as the sex pilus from one bacterial cell to another recipient cell. The other kind of pili is the one that enable the bacteria to anchor themselves to surfaces. Fimbriated bacteria are able to cause diseases such as urethritis and cystitis, whereas non-fimbriated strains are unable to attach to cell surface to cause infection. Endospores are formed by a few bacteria. They form a thick-walled spores as a means of survival when their moisture or nutrient supply is low. So endospores enable bacteria to survive in adverse conditions such as temperature extremes, desiccations, and lack of nutrients. Bacterial spores are referred to as endospores, and the process by which they are formed is called sporulation. During sporulation, a copy of the chromosome and some of the surrounding cytoplasm becomes enclosed in several thick protein coats. Spores are resistant to heat, cold, drying, and most chemicals, so spores have been shown to survive for many years. But when the dried spore lands on a moist, nutrient-rich surface, it will germinate and a new vegetative bacterial cell will emerge. These are cells capable of growing and dividing. The genera of bacteria that are very important medically are the Bacillus and Clostridium. These are the infections that are hard to treat. Examples are infection caused by the Clostridium tetani, causing the tetanus, and another example is Clostridium difficile bacteria that produces diarrheal diseases. This is a summary between a eukaryotic and the prokaryotic cells that we have discussed earlier. Prokaryotic cell reproduction is quite simple when compared with eukaryotic cell division. Prokaryotic cells produced by a process known as the binary fission, in which one cell splits in half to become two daughter cells. Before a prokaryotic cell can divide in half, its chromosome must be duplicated so that each daughter cell will possess the same genetic information as the parent cell. The generation time is the time it takes for binary fission to occur. So, each bacterial species varies from one another when it comes to generation time. On the other hand, eukaryotic cells reproduce in a process called mitosis. So mitosis is a type of division that gives rise to daughter cells for the purpose of tissue growth, regeneration, or asexual reproduction or vegetative reproduction. Taxonomy is one of the topics that we have to be familiar of. So according to Burgess' Manual of Systematic Bacteriology, taxonomy is the science of classification of living organisms. This consists of three separate but interrelated ideas. These are the classification, nomenclature, and identification. Classification is the arrangement of organisms into taxonomic groups on the basis of similarities or relationships. The taxa include the kingdoms or domains, divisions or phyla, classes, orders, families, genera, and species. On the other hand, nomenclature is the assignment of names to the various taxa according to the international rules. Third, the identification is the process of determining whether an isolate belongs to one of the established or named taxa or represents a previously unidentified species. The science of taxonomy was established based on the binomial system of nomenclature. It was developed in the 18th century by the Swedish scientist Carolus Linnaeus. In the binomial system, each organism is given two names. The first name is the genus and the second name is the specific epithet. To express the genus, capitalize the first letter of the word and underline or italicize the whole word. For example, Escherichia coli. It is written like this. In 1969, Robert Whittaker proposed a five-kingdom system of classification in which all organisms are placed into five kingdoms. The bacteria and archaea are in the kingdom Prokaryote or Monera. Algae and protozoa are in the kingdom Protista. Fungi are in the kingdom Fungi. Plants are in the kingdom Plantae. Animals are in the kingdom Animalia. In 1978, Carl Woese proposed elevating the three cell types to a level above kingdom called domain. The three-domain system of classification is based on differences in the structure of certain rRNA molecules among organisms in the three domains. These are classified into three types: the Archaeobacteria, the Eubacteria, or also known as the true bacteria, and the Eukarya, which consists of kingdom Protista, kingdom Plantae, kingdom Fungi, and kingdom Animalia. That's it for cell structure and taxonomy. Thank you for listening.